See, in our previous class we all were dealing with cloning vector. Okay, now here we were dealing with antibiotics resistant gene. Now what we had studied in our previous class, that when the antibiotic resistant gene is inserted within the plasmid, then for example two type of antibiotics are there, that is tetracycline resistant gene. You see, tetracycline resistant gene is used. So the recombinant plasmid means the plasmid that from which the DNA has been inserted, now will lose the tetracycline resistance. Okay, now due to the insertion of the foreign DNA, it will lose the tetracycline resistance because of the insertion of foreign DNA, and it will be selected out. Now how will you select? It will be selected out from the recombinant, non-recombinant one by plating. How will you do? By the plating the transformant containing the medium. Okay, this was there because see, if two different one, the recombinant one will grow in ampicillin containing medium, because it was resistant to tetracycline, so it will grow in ampicillin, but the non-recombinant will not grow in this medium. It will grow in both the medium. Okay, recombinant will grow in ampicillin but not grow in tetracycline, and the non-recombinant will be growing in both. So this is how you can, and you can select the recombinant one. See, selection of the due to the inactivation of the antibiotics, and this is a very very primitive, and it is a very cumbersome, that means it is laborious and time taking. So what is the new technique? New technique is the separation of the recombinant and the non-recombinant on the ability to produce color. See, produce color in the presence of a chromogenic substance. What is chromogenic substance? The substrate that imparts a color. Now, what is the procedure? Now, here you see the recombinant DNA. See, the recombinant DNA is inserted within the coding sequence of enzyming B-galactosidase. So, the beta-galactosidase, here the recombinant DNA is introduced here. Now as a result, what happened? The inactivation of the gene for the synthesis of this enzyme beta-galactosidase, so it won't synthesize the enzyme. So what and this particular enzyme, see, we are using a chromogenic substrate. Now here for example, this is a DNA plasmid. Okay, this is a and here we are using insertional inactivation. So here the insertional inactivation, we are using beta-galactosidase. So now see, this is a red medium, that means this is a chromogenic substrate medium. So this plasmid, when it is number of plasmids are there, now you see suddenly few turns red, group of plasmids turns red and other do not change color. So those that turns red, that means it does not have the insertional inactivating enzyme, and here this portion they don't change color. That means what? Here these plasmids, the recombinant one, have been inserted by this gene, so it will not change color. So this is a very easy technique of separating them. Okay, now see, in nature, if we take off nature, see, in nature from a long time we have been using this vector cloning. You see, vectors for cloning gene in plants and animals, it have been used for so many times. Now for example, see, for example here we have Agrobacterium tumeficans. What is this? It is a pathogen. Okay, pathogen for several dicot plant, but it is able to deliver a piece of DNA, that is known as tDNA, to transform normal plant cell into a tumor. So if this tumor is detected, this new tumor cell to produce chemical required for the pathogen. So this is a very natural way where plant breeders or genetic engineers use it to infect this normal cell with the tumor. So when the tumor will burst, this bacteria that produces pathogen will provide chemical required by the thing. So this is tDNA will be introduced to the pathogen. So and here in case of adenovirus, you see there are certain carcinogenous this thing, cancerous cell. For example, see normal cell, if some cancerous or carcinogenic substance is introduced within the cell, that normal cell, that is known as proto-oncogene, gets transferred into oncogene, that is we have already read. So this is also a very common way where you can transform the normal cell into a cancerous cell, and then it can multiply, where it can multiply. Now another thing that we will be dealing is, see, when we want to introduce a particular piece of DNA into a bacterial DNA, that the bacterial cell should be competent enough. Now what is competent? What does this word competent? Competent means it should be suitable. So how is it done? Now here see. Now see again here, this is the competent host. Competent host for transformation of recombinant DNA. What happens? See, DNA chemically it is hydrophilic in nature. What is hydrophilic in nature? It is fluid loving. And if we simply reconsider the case of the cell membrane, now see, cell membrane, it's hydrophobic to some extent. So this DNA under normal condition will not enter the cell. So it is very difficult to pass them through the cell membrane. See, cell membrane, it is, so what has to be done? The cells are to be treated with a specific concentration of divalent cations like calcium. Okay, if calcium is given more, so that will increase the cell permeability of which cell? The bacterial cell, into which we want to introduce the DNA. Now you see, simply if you treat it with calcium, it would not increase the permeability, cell permeability. So what you have to do? You have to provide heat shock. Now how to provide heat shock? It has to be heated up to 42 degrees centigrade and then placed on ice. So this is how the cell, and after that when again this DNA is introduced, it will simply get absorbed into that. Another very common example is micro injection. What happens in micro injection? Simply the DNA is injected into the host cell. So the normal extra chromosomal DNA will take in this. And another one is known as biolistics or gene gun. So see, biolistics and gene gun, you know, it is only found in plant cell. This technique cannot be used with the animal cell. And what happens, you know, this gold coated, see, the DNA is coated with gold and tungsten, these two metal, and then it is bombarded into the cell with high speed. So when it is bombarded into the high speed, it is normally injected within the cell type. It is similar to micro injection. And the lastly, another method is disarmed pathogen vector. What is disarmed pathogenic vector? Actually, the cell has to be infected with the pathogen to let the DNA get introduced within the cell. So this is what we are having in competent host. That means the host cell should be suitable enough to take up the recombinant DNA. Now you can see in your book also, see the DNA, it is hydrophilic in nature. So it has to be, see, divalent cation, specific concentration of divalent cation, that calcium, that increases the efficiency through which the DNA enters. And here you see bacterium pores, see, heat shock is also mentioned. 42 degree centigrade it has to be heated, and then again it has to put back to ice. So this is how it has to be done. Then you have the micro injection. Now what is the micro injections? The recombinant DNA is injected directly into the cell molecule. Then another one, last what I was telling was biologistic or gene gun. Here the DNA is coated with gold and tungsten and directly it is bombarded. That means it is with a great speed injected into the cell or thrown with a great velocity into the cell, so that the gold and the tungsten that pierces the membrane and enters the DNA, and once it enters the DNA, it mixes with the recombinant, it forms the recombinant DNA into the host. And the last one is, you see, last one what I said is, see, disarmed pathogen vector when it allows the DNA to infect the host cell, and after infection it transfers the recombinant DNA into the host. So these are the things, and main and primary form of introducing the DNA into the cell after it has been selected. Earlier when we were dealing with cloning vectors, we were dealing with how it is the recombinant DNA is selected from the medium after it is cut. Okay, and here how it is introduced within the cell. In the next class we will be dealing with process of recombinant DNA technology. We will be dealing with the processes.